Wolfman.
You're listening to UCY.TV. Why don't you stick around for more awesome radio?
All right, good morning, everybody. Today is Tuesday, May 17th, 2016. Don't push it, Joel. And this says, before the first cup with Jules here on UCY.TV, happy Tuesday, everybody, in 2016. It is 51 degrees and overcast here in upstate New York. Looks like we're going to get some rain. Seeing it's all down over Pennsylvania, New York, and New Jersey. Not quite here yet, but I have to say, I feel very nasally challenged with pollen in the air everywhere. This has been a rough couple days with sniffly noses, but hopefully when all these flowers are done, I will be recovering a little bit. So where do we leave off yesterday? We titled the show Daddy's Beer, Da Vinci's DNA, and a Fully Synthetic Human Genome. And that's just about puts it in a nutshell. We talked about how parents, especially dads, and this was not something that they were fully aware of before, things that the dad did even long before conception affected the child. I mean, that just really makes sense. It's common sense that if you ingested some stuff that's going to make you a defective human, that those defects would be passed down to the defective human's offspring. And that would include drinking beer. No, I think they said alcoholic, which, they had always kind of guessed that alcoholic genes were passed down in the family, but they said it could cause mood issues and it could cause immune issues, and there were all kinds of other things. But for the mother as well, I just saw, I thought that was right here. Moms can transmit psychiatric trouble to kids, according to a study. Let's see. Individual symptoms such as anxiety, avoidance, and heightened response to stress can be transmitted from mother to child and even to grandchildren by non-genetic mechanisms. A new study by investigators at Weill Cornell Medicine and other institutions show preclinical findings published May 13th in Nature Communications may lead to tools to predict if a child is at risk of developing behavioral problems later in life after exposure to stress signals in the womb. Genetic and non-genetic inheritance are different but complementary, and this might be these epigenetics that we were talking about yesterday as well. Your parents and grandparents' experiences can literally be passed down to multiple generations of offspring. The one big study that they've been using is, we read they were doing studies in Tel Aviv at Tel Aviv University on Holocaust survivors in regards to psychological stress. I don't know, I saw after the show that there were a bunch of stories after that were doubting the link because it was only just a small study sample that they were following those epigenetic changes in. But I could tell you, I could absolutely see how it's possible. It's called survival of the fittest. That's what biology is all about. It's live and survive. So, if there was something that was majorly traumatic, of course it's going to get encoded into your sequence to be passed down. That's how the information is passed down to make sure the next generation makes it to be able to reproduce. So anyway, I don't want to dwell on this, though, today. So let's see what else we looked at. I'm going to talk about a few things today. We also talked about Da Vinci's DNA. They are looking, and, it's kind of a pipe dream right now, but they want to get a bunch of people together, including Craig Ventner. They have Queen Elizabeth and Billy Gates, who actually own some of Leonardo da Vinci's writings. They want to be able to get some DNA off of one of the items that he owned at some point in the past and be able to create synthetic genomes, and specifically a synthetic genome for Leonardo da Vinci. And, we did two stories yesterday. There was the Leonardo story, being able to sequence him, and then another one out of MIT Technology Review about now they were, actually it was out of that was out of New York Times for the synthetic human. They had a big meeting at Harvard with 150 scientists talking about how and what and if they should sequence all 3 million DNA sequences, since they already know all the chemicals that go into making up each of those CATGs. They just need to know the sequences now. All the different permutations that can happen, and put those into a database so then they can push a button and pull a trait based upon a sequence. They already created all the chemicals, right, that they could then spin up into whatever select traits they want and build a human without ever having human parents. I found another story because the New York Times article kind of blibbered and blabbed, just like a good New York Times article will. That's why the New York Times Sunday edition is so huge. But ScienceAlert.com, which is a publication out of Australia, these guys usually do a good kind of bake down. So let's read this one, because this is the same thing on creating the synthetic human genome, to kind of outline a couple things that we never heard them mention in the New York Times story. This one is titled 150 Scientists Just Met in Secret to Discuss Creating a Synthetic Human Genome. A very select group of scientists, lawyers, industry leaders just met at Harvard Medical School in Boston to contemplate building an entirely synthetic human genome. That means figuring out which chemicals are needed to create the 3 billion, it's billion, excuse me, I said million. The 3 billion bases of DNA that sit inside the 23 pairs of chromosomes found inside every cell nucleus in our body. Journalists were barred from the meeting, and participants were not allowed to tweet during the proceedings, both of which have raised suspicions that the ethical considerations involved in such a project are being overlooked. The proposed project has been described as an unofficial follow-up to the Human Genome Project, which saw thousands of scientists around the world read and record entire sequences of the human genome. And in the largest international collaboration ever taken in biology, that project ran for 13 years, finally completing the human genome on April 13, 2003. And who would have thought? I remember when that happened. And who would have thought in 2003 that we would be sitting where we are today with mad science exploded in our face? Beyond anything, I just heard some other host somewhere on a much bigger show say what I've been saying, and I think they listen. But saying exactly what I've been saying, like in my wildest dystopian nightmare, did I never see some of the insanity that we are seeing now. Maybe I just wasn't creative enough because I guess it was all possible, but on a daily basis, when I go through this news, there's red button issues everywhere because everything that you question, every little thread that you tug on, it starts to unravel into one big ball of crazy insanity that nobody's thinking about, that no one's thinking about in multiple generations from now, because you know what? Biology doesn't matter anymore. It's all about synthetic. And so let's get back to the synthetic genome. So in 2003, it's when they first completed the 13-year project to sequence the human genome. And now this is going to be considered part 2, creating the synthetic genome. So now scientists want to do the whole thing again. But this time, they're not reading the human genome. They're writing it. The project is called HGP Write, testing large synthetic genomes in cells, aims to synthesize a complete human genome in a cell line within 10 years, which means replacing the human genome in human cells with an entirely synthetic one. Wait, let me stop. I'm going to read that again. The project, called HGP Write, space, testing large synthetic genomes in cells, comma, so that's the whole project name, aims to, quote, synthesize a complete human genome in a cell line within a period of 10 years, unquote, hyphen, which means replacing the natural genome in a human cell with an entirely synthetic one. Now let that sink in for a minute. Sucking out everything that makes it alive and replacing it with something that was created by a human in a lab that isn't alive, out of chemical substances that you synthesize and put the ingredients into a database. How absolutely horrifyingly frightening. You know, they kind of quipped in that New York Times story yesterday. I don't know if they mentioned Leonardo in that one, but I know they mentioned Einstein, and they were saying, you know, well, what can we do with this? Maybe someday we get Einstein's genome and clone him. And maybe, and I think it was the Leonardo story, though. Leonardo story, they're talking about, hey, maybe we clone Leonardo. And I was joking, you know, we get 100 of them and stick them around at all the best art schools around the world so you could be a teacher. But what, I mean, we used to kind of laugh at, oh, damn, what was the show with Bender? Bender the robot, where they used to have all the heads. You know, they had the heads of all the famous people in the jars in the rooms, and they could all talk to you in the future. Futurama, that's what it is. Oh no, I lost my train of thought. Oh, cloning. You know, so here we are, play things like Futurama, where we always kind of envisioned heads in jars, and I think that sort of came from the concept of suspended animation and being able to be cryogenically frozen, right? They always assume that if they froze your head, that sometime in the future they'll be able to bring you back. Well, now they're finding out, well, might not be the case. I mean, yeah, we can get DNA, but you probably should have kept the whole body. So we could have used some stem cells. We don't just need your head. But now they're talking about being able to get your DNA in Leonardo's case from a manuscript that he wrote hundreds and hundreds of years ago, that's now in some rich, very rich, the elitist, richest people's possession, and use some scanning technique with lasers to find where the DNA strands are, and then potentially use those to create a new copy of Leonardo. Or they mentioned Einstein as well. Tweaking them any which way. And once we learn what each of the ATCG combinations and soon to be X and Y, maybe we'll get into that today. Some of the two synthetic proteins or aminos that they've added now that they want to mess with, and they probably already are in the lab. We know they are. But what kind of beings are going to be created? Superhumans. And then even more disturbing is the fact that Bill and Melinda Gates, of course, have their big stinky toe stuck in the synthetic biotics. We looked at that Synlogics after we found what a direct connect we have between our gut flora, our immune system, our moods, our brains, our whole bodily harmony are involved with having good gut flora. And Bill and Melinda Gates are now invested in an MIT spinoff that can program that bacteria to do just about anything they want. Now, I just saw I have so many stories open right now, but I just saw a story that some of this new stuff that they're working on, oh, it's this one, is going to completely make CRISPR and CRISPR-Cas9 obsolete. So, I don't know, when I saw that, and we'll get down there, but when I saw that, I mean, even though this is beyond horrifying, if this puts CRISPR out of the picture, that means at least those 60, and I mean, it might still be able to be done at a DIY level. But remember that lab was sending out CRISPR to whoever wanted it to be able to slice and dice genes in your living room. All right, here, let's read the rest of the story about the synthetic human genome. So within 10 years, it means replacing the natural genome in a human cell with an entirely synthetic one. Of course, a closed-door meeting about cooking up the blueprint of life in a petri dish was always going to raise suspicions, with the possibility of using synthetic human genomes to build Bourne-style super soldiers. Ding, ding, ding. Didn't I say that yesterday? And I know it sounds crazy. I know it sounds like an X-Files episode, and I'm not talking the new ones. I'm talking the ones from the 90s. But DARPA, you know, our nemesis here on this show, DARPA's been working on that for a long time, and it all goes along with being able to manipulate, control, and understand the brain and the body, putting you on a remote control and genetically engineering you to be the beast that you deserve to be. to be their slave, their cannon fodder. At least if you're created in a petri dish or however they're creating these synthetic genomes and storing them and then developing synthetic embryos out of them, I would imagine that they wouldn't feel too bad about blowing up replicants. The problem is, once they start creating replicants, we're going to run into a whole bunch of trouble. Because not only will they probably program these things, especially if they're super soldiers, with no emotions, they'll be able to maybe do lie detection and stuff, but they're not going to have any type of empathy. Super soldiers are going to be killing machines. That's what they're going to be. Synthetic killing machines. And if they were created in the lab, they probably were given superhuman traits. We joke, but wings, claws, bug pinchers, x-ray vision. What kind of a world are we leaving? And the people who are doing this aren't thinking about what kind of a world are they leaving, because their plan is within 100 years, they're gone, man. They're heading to the stars. And they got their eyes on all kinds of exoplanet candidates now. I just saw something yesterday that they can even, they discovered you could take an icy dead planet and with a supernova or some sort of other cosmic explosion, it could actually terraform a dead icy planet into something living. And we already saw the DARPA lady talking about that last year in that Motherboard magazine article, saying that that's their hope. Synthetic biology, that is their hope. To be able to push a button, pick genetic traits, slice and dice from across the animal and human kingdom. So not just humans. We're talking mixing with bugs and different animals as well as human DNA to make something different. But she also said that their eventual hope is to use some of the synthetic biology to terraform Mars. Well, that's already what we found out. We found out that NASA is planning on abandoning the space station. International Space Station, after all these years and all that money, yep, we need to move on to something bigger and better. And so what it's going to start with is moon base. Moon base for short hop, skip, and a jump to move supplies. But it's going to be the launching point then to Mars. And we already have probes. It's been quite a number of years now. Curiosity and I remember the other one, the two rovers that are still zooming around. And then they got all those satellites that are mapping the entire Martian surface for all the minerals, of course, to show where all the good mining will be and where you can make lots of money. But they literally are planning on a Mars colony. And who knows whether or not it's actually going to be the reality TV establishment that they had talked about, where they had all these people sign up. I don't remember how many there were, but they had millions of applications from all over the world for people to go on a one-way trip to be on a reality TV show called Mars. Get that all established and start terraforming it so we could have some oxygen and trees. But the plan of the rich people is not to stay on Earth. So if they release an army of minotaurs to come eviscerate every single one of the last remaining puny humans, it's not going to matter to any of them. They don't care. They're off to exciting new places with their exciting new synthetic bodies and synthetic DNA so they can live forever because, hey, they cured death as a disease. See where this is going? But like I said, we're the guinea pigs to get there. We're the jumping leapfrog. But it's not for us. They're not going to pack you on a ship and take you with them. That's fine. I don't want to go to Mars. I don't want to go to Alpha Centauri like Stephen Hawking is planning. Those little mini probes they're sending out. I want to stay here on Earth. I like it here. But I want them to stop screwing with our planet. I mean, once they have completely bastardized, and what happens when they create creatures that can't be killed? What happens when they create creatures that have full regenerative capabilities of all their limbs and brain and body parts? So even if you hack the head off a Hydra, guess what happens? Grows back another head. I just saw recently they cracked some of the secrets of the one lizard. I don't know if it was a gecko. Don't quote me. But it was, I don't know if it was one lizard in particular, but it was the lizard genes that allows them to grow back limbs. And they had just figured out how that all happened, and now they're sequencing that and hoping to be able to use it in humans someday, right? Well, imagine doing that on a grand scale in an organism so everything regenerates itself. Then you couldn't die. Then it would make you immortal. So if you had superpowers, or let's say the super soldiers that they want to, born style super soldiers that they want to create, and if they made them so they could just keep reproducing limbs and body parts and cells every time they got damaged, and put superhuman traits in them, these would be like demons from the pits of hell, guys. That would be in the pits of hell for eternity. It's very, very, very frightening as they shoot off to the stars. This literally is a battle for the survival of human 1.0. Anyway, back to this. So, of course, a closed-door meeting about cooking up a blueprint of life in a petri dish is always going to raise some suspicions. But with the possibility of using a synthetic human genome to build born-style super soldiers and countless Einstein or Leonardo da Vinci clones in the future being not entirely outside the realm of possibility. And I know anybody who's already left now because they're going, man, just talk about X-Files and super soldiers. This chick is completely nuts. I'm not nuts. They are talking about this. There are official projects going on. This isn't hearsay. This isn't lunacy. I will guarantee you in a back room in China, they already have a bunch of clone pods full of special clones. The ones that they're going to make their Chinese army with. Why do you think they built a clone factory that could pump out 1 million units a year? Wasn't to do cows, like they said. I mean, maybe. Sure, Bessie. 1 million Bessies. But the clone factories to clone people. Why do you think he came out and said that? Or little Fido for $50,000 a pop. I just saw too. See, China doesn't give two shits about their people. And here's something I saw. They now are looking. We've talked about the replacement of jobs with robots. And I probably saw 4 stories at least just in my little evening foray last night after shows ended before I went to bed. 4 stories about more robot stories taking jobs, lawyers being chucked out now. But in China, we talked about Samsung in South Korea building automation robots specifically to send over to displace the poor people working in the factories in China. Well, I saw now, there was a story yesterday about them and how they've been using, and a lot of that I've seen in Japan, but I guess in China as well, where they've been doing, and they're doing it here now too, automated restaurants, right? Get rid of all the minimum wage employees. We don't care. Let's make it all automated and have robots deliver food. Well, people didn't like the robotic look, and we've done that too, the whole aesthetics of how humans embrace technology, and especially something like a helper bot that would be around if we give it to Nana to go help her, roll over and not get bed sores. So they discovered in China that, yeah, we need more android-like robots to be able to replace all the humans. We might have to look at that story later. There was a video that went with that. But, guys, it is coming. So anybody who looks at this and says, Jules is a lunatic and none of this is happening, and oh, my God, she's so full of fancy. You know what? I think you need to go get your head checked. Start doing some research. If you spend as much time as I did, and I don't look at Billy Joe Bob's blog, okay? The second I don't recognize a source, I don't read it because it's a waste of my time. Half the time, most of those people don't source the material. I try to go to the research journals, to the FBO.gov, where DARPA's requesting requests for proposals, and the actual black and white letters are printed on paper or, simulated electronic paper. But I want to see it for myself. Don't believe it because I tell you. Don't believe it because someone else said it on the radio. Go look it up yourself. I heard Hal saying this the other day. And don't read the article on it. If you see that there's a link or if you can find the raw document, don't take what the author wrote about the document as what it meant. Read it yourself and come up with your own interpretation. And then come to me and tell me that this isn't happening. Because I will guarantee you that there's nothing that you will find that'll prove me wrong. Anyway, so yes, closed door meetings raising suspicion with the possibility of using synthetic human genomes to build Bourne-style super soldiers. And there's a new Bourne movie coming out. I saw. I know, don't watch movies, but if you ever saw the whole Bourne series with Matt Damon, it's the first time I'd ever really paid attention to Matt Damon as an actor. And I got to tell you, those Bourne movies, talk about exciting. Those are some really, really good flicks. But yeah, I guess there's another one coming. So talk about pre-programming, Bourne-style super soldiers, and you would also be able to create countless Einsteins or Leonardo da Vinci clones. And that's what we talked about in the future being not entirely out of the realm of possibility. Would it be okay, for example, to sequence and then synthesize Einstein's genome? Lori Zoloff, a bioethicist at Northwestern University, and Drew Endy, a bioengineer, in Stanford University. That's where DARPA came out of. That's where a lot of the stuff came out of, was Stanford. Wrote in an essay for Cosmos magazine criticizing the Nassian project. Somebody from Stanford is criticizing this. It's pretty interesting, too. So if it's okay to go clone all these long dead people after creating synthetic genomes of them, after DNA sequencing them. So look, they're working with Craig Venter, the guy who first did sequencing on bacteria. He's got himself a big sequencing company, and he's an extremely rich bazillionaire. Venter is actually working on both the projects, right? The synthetic human genome and the Leonardo da Vinci project. How convenient. So let's see if we can find somewhere along the way. We got an Einstein coffee cup or an Einstein sweater. I'm sure it's out there. We find there's a hair on it. We take that hair to Craig Venter. Craig Venter DNA sequences it. Finds out the sequences, ATCG, all spinning in the double helix. They're able then, because they already know what's in each of those chemical elements, let's synthesize all the chemicals. And now, laser 3D print that DNA sequence. Remember the 32-year-old rich kid that created his laser 3D company, Cambrian RNA or DNA, Cambrian something it was called, who ended up with an untimely demise just recently we read about. Oh, they could very easily do that now. Laser 3D print Einstein's genetic code. And it would be a synthetic genetic code if they sequenced that piece of hair and then reproduced that DNA. So they didn't actually take the DNA from the hair and get it to reproduce. They would take the DNA sequencing from the hair or the skin or whatever and recreate it utilizing synthetic means. The elements don't need to be synthetic, but the fact that we are synthesizing proteins, aminos, to make the sequence from its raw materials, mind you. So this is kind of like that desktop manufacturing type stuff, right? We could go to the raw material level of creating matter. And as long as you know the correct sequence or the way things all go together and the shape and the form and the way everything fits, you can create anything, right? We're about at that level with technology now. We are gods. So they'll recreate Einstein's genome and then they clone it. And Einstein gets born again. I don't know what they would put that embryo into. Maybe an artificial womb, right? They're working on those too. Could have an entire lab full of artificial wombs with synthetic genome created embryos. You didn't even have to fertilize with a real sperm cell because you can create those too. Synthetic egg and sperm. Till it grows into a cloned copy, albeit with synthetic DNA, but an exact duplicate of Einstein's DNA. You know, and they're supposed to be discussing the ethical implications of this. There's no ethics involved here. You can't just, going back to Futurama with the room full of heads. This is like 1,000 steps beyond that. So what, George Washington? Just walk into a room, you got George Washington. We got a George Washington clone. Fast forward 30 years into the future. George Washington clones at every American History Museum to sit and talk to the kids when they bring them in for the school field trip. Like I said, we got Leonardo da Vinci's at all the best art colleges and engineering schools for the kids that can pay, doing tutorship and teaching. If so, anyway, so this engineer at Stanford University says how many Einstein genomes should be made and installed in cells, and who would get to make them? That is a big question. And he's criticizing this project. Unlike conventional cloning, which requires source DNA from the original organism, like Dolly the sheep, and we talked all about that. And remember, one of the issues that they ran into, and there was still question as to whether or not cloning, in Dolly they thought they ran into it, but then I found conflicting data, so they've never proved this, I don't think. Then in cloning they had issues, especially in sheep. Not in cows, though, but sheep were an issue. Where when they clone them, it shortened the telomere. And we know already from all this longevity medicine we've dug into that they've discovered that that telomere is one of the key items that causes aging. When those telomere ends start to shorten, and those can be affected by your lifestyle, lack of sleep, booze, sugar, all the shit you eat and do that isn't good for you. Those shorten your telomere ends. And as those telomere ends shorten, you get older and older. And that scientist back a few weeks ago, that woman CEO, who just injected herself with, I don't even remember what it was. Remember, she used herself as a guinea pig. Some sort of telomere lengthening genetic modification that she made to herself using CRISPR or whatever. She says, I'm feeling great. But the telomere length was an issue in cloning, and Dolly they had said that Dolly died early, but then more scientists came out later and said, no, Dolly didn't die early, so who knows? But they're not finding the telomere issue in any of the other clones that I saw. When we dug deep, we did, I don't know, a week or so on clones back the end of last year. Anyway, unlike conventional cloning, which requires source DNA from the original organism, i.e., think Dolly the sheep, creating a clone from synthetic DNA would require no natural source. You just need to cook up the right chemical cocktail based on the original recipe. And this is like making counterfeit Kentucky Fried Chicken. Except in the likeness of Einstein. Andy, this is this Andy from Stanford, was invited to the meeting at Harvard, but he declined amid concerns that it was not open to enough people. Discussions to synthesize for the first time a human genome should not occur in closed rooms, says Andy Ann Zoloff, the other researcher that published the paper questioning how smart this is. Harvard geneticist George Church, one of the organizers of the meeting, told Andrew Pollack at the New York Times, and that is the story that we read yesterday, that there's been a misunderstanding about the intent of this proposed project, saying that it's aimed at creating synthetic cells, not synthetic humans. Of course not. And would span many life forms. So, hey, humans aren't exclusive, including all kinds of animals, plants, and microbes. I mean, we might as well spread the wealth of insanity. We might as well spread the apocalypse to everything that lives on this planet. You've misunderstood. It was not just for humans. And they're painting, a picture which I don't think represents the project, says George Church, adding that the meeting at Harvard was closed off and attendees were told not to tweet because the organizers had submitted a paper, blah, blah, blah. Yeah, he says he didn't, while it's going through peer review, he didn't want any secrets released. So just make sure you keep it on the QT till it's all reviewed and then we release it. So whatever, maybe not, but I would think that if you were having a meeting on something as big as synthesizing a synthetic genome of people like Leonardo da Vinci and Einstein and creating completely synthetic DNA, regardless if it was human or not, that should be something that's open to all scientists and maybe even to all global citizens. You know, I don't like to be considered like part of this big global UN conglomerate where they're going to be able to stomp their feet on us and put universal rules against us. But I would say as the human race, you know, like Reagan had said that if we had found ourselves at threat from an outside force outside of this planet, you know, we might all pull together in unison to protect the human race. Well, I think we're all being threatened as a human race from our own. From these elite lunatics that are doing things like trying to completely displace biology because it's defective. Because we feel we have the right to create and modify life in our own image now, or with our own fantasy, or with whatever traits that money can buy. I think as global humans, we should all be stopping this. You know, this is going to be sold to us as our cures for everything. But eventually, it's not going to be 21st century medicine. Right now, it's little nanobodies or little program bacteria, or, you know, we're going to send payloads in and blow up your cancer tumor, and all targeted therapies would be great. Tomorrow, the fix is going to be giving you your shot of your synthetic DNA sequencing in your arm. I don't know. I mean, this is just, it's so difficult to wrap your mind around this. You're going to regrow a synthetic limb. It won't even maybe be prosthetics tomorrow. And eventually, you know, when you die, anybody ever watched I watched both, the original in the 70s or 80s and then the one in the 2000s, the Battlestar Galactica. When those skin jobs got killed in Battlestar Galactica, and those were the robots that then became artificially intelligent that then evolved into human S-type robots. And then when they died, they just re-uploaded their consciousness and bloop, another one woke up. That's pretty much at the point we're at right now. They've been messing with all those technologies. We have Kurzweil wanting to upload yourself to the machine. You could download Nana at Christmas time in her avatar. I really think it's a possibility. And now if we can have a fully synthetic human genome, guys, those are called skin jobs. Get that? Even more than skin jobs. I don't even know what to say. This has to be stopped. This is the destruction of everything on Earth is happening right in front of our eyes. All in the name of science. All in the name of look at how great this is, and we're going to do it because we can. And screw you. We're going to have a private meeting. You don't need to know about it. We don't need to tell the whole world what we're doing or need your permission to do it, except it affects every single one of us. You know, these experiments, and there's so many of them. I mean, any of them. Anything that we are uncovering, any type of conspiracy, any type of poisoning, whether it be pollution, nuclear, chemical, biological, wars. You and I didn't vote for that. We didn't wake up in the morning and go, let's go drop bombs on lots of innocent children and women in the Middle East and kill them all because, I don't know, because there might be a boogeyman in their closet. None of us woke up and wanted to do that yesterday morning. But they are orchestrating the destruction of not just the planet now, but of the human genome. And just like we have the seed vault up in Iceland to protect seeds, which I really, that's been up there for quite a long time now. When I start thinking about how genetic modification of plants has gone completely off the rails and Monsanto and their poisoning, I mean, we know now that a lot of what's going on is what is getting into our gut through our food, through these pesticides and modifications. It's what's screwing up, the human immune system and all the other issues that we're seeing. I mean, we've done quite a few shows on that. That seed vault is storing heritage seeds, seeds that haven't been modified, seeds that have not been germinated, utilizing faulty, genetically modified pollen. They're safe seeds. They're the seeds that they're going to pack up when they go to Mars and start all over again, or Alpha Centauri. Well, they're now polluting and have already polluted between the vaccinations and the foods and everything else, the human genome as well. And I bet you if you went back just 100 years ago and you look at what the human genome looked like then compared to now, you would find that there were some disturbing changes. We listened to Dr. Mercola talking to that Dr. Perlmutter last week in regards to gut bacteria and a lot of the changes that they've seen. And he was saying that our genome now, and it went for thousands of years, the human genome, without ever changing at all. It didn't change. It stayed very stable. And here, just in the recent past, there are these significant changes happening. And he was saying that because of all this poison that's being put into our environment and our body, there are changes happening where switches are getting flipped on and off in our DNA in reaction to some of these environmental factors now that are happening. And our DNA is changing faster than it ever has in thousands. I had said millions, but it was like thousands or tens of thousands of years. And they found that by being able to go back. I just saw it was like 434,000 years old. They seek DNA sequence. We read that yesterday. Some human material. So they have records back quite a long time to be able to see where we were at and where we're going. All right, I'm only going to be able to, if I look at the time here, I'm not going to get very far. So let's finish the story because I want to get to a little bit more. Anyway, so we're talking about sequencing. Da Vinci, Einstein, making super soldiers. And they're all talking about this at Harvard. Anyway, this church dude that's in charge of it, he's like, no, it's a misunderstanding. We're not looking to do that. He says they are painting a picture. He says it's not just for humans. We're going to do this modification, this synthetic DNA, synthetic genome project for many life forms, including animals, plants, and microbes, as well as humans, because that's what the DARPA lady told us. Yeah. They're painting a picture, he says, which I don't think represents the project, adding that the meeting was closed off because of their journal thing. And he and other organizers said that ethical aspects have already been amply discussed since the beginning. Well, what the hell then did they decide on that? Because I'd like to read your ethical papers. And what your plan is there, church. The project has not yet been funded. But, hey, don't worry about it, because there's lots of rich tech billionaires that would love to be able to control all of DNA and own it synthetically at the push of a button to create any life form they wanted. So don't worry about it, Johnny, because you're going to get your funding. But Church and his team have approached various companies, foundations, and U.S. government to ask for support, and I'm sure DARPA will step right up. If they do raise the capital to launch this thing, then they got a tough road ahead. Remember, they also got Craig Ventner, billionaire, that's right in the middle of all this. Because even though it took more than a decade to get it right, reading DNA is a whole lot easier than actually writing it. And right now, synthesizing DNA is difficult and error-prone. Existing techniques can reliably make strands that are only about 200 base pairs long, with the base pair being chemical units in DNA, and a single gene can be hundreds, if not thousands, of base pairs long. And to synthesize one of those, multiple 200-unit segments have to be spliced together. But consider the cost of synthesizing genes has gone from $4 a base pair in 2003 just to 3 cents today. We could see the entire 3 billion base pairs that make up the human genome drop to as low as $100,000 in just 2 decades. That's what it would cost. That's what it would cost to sequence all 3 billion base pairs that make up any human that has ever existed on planet Earth and put it in a database. Very shortly, that's going to cost you $100,000 to own all of that, to be able to make any of your own life. If this project is a success, all that genetic tweaking that we're seeing, and listen to this. So if this synthetic human genome project is a success, all the genetic tweaking that we're seeing from the new and much touted CRISPR-Cas9 gene editing method, in which scientists have recently used it to modify human embryos, create yeast that produces insulin, and edit HIV out of human cells, even though it later returned, could be made completely obsolete. This technology is right, you wouldn't have to edit anything anymore. You could just create a new DNA sequence. Instead of tweaking what nature has made, like we're doing with CRISPR-Cas9, hey, Clyde, we could just build it ourselves now, out of the database, with the touch of a button, or even a smartphone app. So, where does everything stand right now? Well, with little information coming out of the May 12th meeting, all we can do is sit tight and wait for that peer-reviewed paper that Church talked about, why he wanted everyone to zip it. We should wait for it to be made public so we can see for ourselves exactly what was discussed, and whether or not the ethical considerations will be given due consideration in the future is anyone's guess. But at this stage, it's more likely than not that this project will go ahead. Of course it will. There's no putting the brakes on any of this mad science. It is like lemmings off a cliff, full speed ahead. Remember, I told you psychopaths get off on the thrill. That's what this is. right at the brick wall. Because at the very last minute, ta-da, Superman flying through the air with his bat wings and, eagle claws to snatch you out of danger's way. He's going to appear at the very last second from their genetically modified, synthetically created super soldier to rescue you at the very end. I mean, this is complete, absolute insanity. Says the results for this could be awesome. Yeah, awesome. We just have to make sure that ethical issues are given as much consideration as what? Oh, the business opportunities. And that is what it all comes down to now, isn't it? It's the business opportunities. How much should it cost? What's human life worth? Should we be replacing humans everywhere? Speaking about heads, Futurama, sticking them in a jar in a room. Remember we talked about that head transplant? There was that Italian surgeon. We talked about him last year. He said he was already able to transplant a head on a dog or something, which the Russians had tried way back when. There's a pretty disturbing research film that I've seen on that. But there's an Italian scientist over the last year or so saying that he has the technology now and the ability to reattach heads. He was looking for his first volunteer to be able to put on a donor body. And originally it was a Russian guy who had a very debilitating disease. It was looked a lot like anyway what Stephen Hawking has, where his brain's all there, smart cookie, but his body is all kind of mangled up and it's just a degenerative disease. And so the Russian guy was the one that was getting ready to be all lined up to be transplanted. Well, listen to this. I guess they changed their mind. Story came out for an update yesterday. World's first head transplant and to now be carried out on a Chinese patient. China paid more money next year. Italian neuroscientist Dr. Sergio Canavero, same dude, has announced that a Chinese patient will receive the world's first head transplant in December of 2017 if the surgeon can receive the funding and approval. Cavanero told the Times of India that technology and the team to perform the surgery is ready, meaning funding and ethical approval are the remaining hurdles to the potentially groundbreaking operation. And I bet you, Putin has stood up and said he's had some morals about lots of things. But in China, it seems to be mad science. Anything goes. They've genetically edited human embryos for a second time now. China's got cloning factories. I don't think that they would put a kibosh on doing a head transplant, especially if they were the first in the world and China could take some credit for it. Anyway, the doctor also optimistically suggested that the patient could be up and walking again within a year of the extraordinary surgery. The Chinese team is already experienced on human cadavers to hone the technology. So he does have Chinese doctors that are getting ready to work with him. Cavanero said his first patient will be Chinese, despite previously suggesting that it would be Valery Spindirov, a Russian tech scientist who suffers from a rare muscle wasting disorder. But Cavanero pointed out that Spindirov could not receive a donor body in China due to biological and ethical reasons. The surgery is inherently high risk, but Cavanero believes that the operation's success lies in cooling both the donor and the recipient down to 12 degrees Celsius or 53 degrees Fahrenheit so the cells do not die of oxygen starvation. And yeah, that's the suspended animation stuff that we've talked about a couple times now. Once you cool it, everything slows down like you're in hibernation mode. And once the head's connected, the patient will be kept in an induced coma for 7 weeks until he fuses back together, and we use electrodes to stimulate the spinal cord. All right, guys, well, we'll continue tomorrow. Make sure you stick around. It's Tuesday. Mr. Kevin Blanche is live up next. New Chuck Ocelli at 3 p.m. Eastern, live at 7. Ron, Game On, 8. Tim Kelly, 9. The boys from Primetime, live and at 10. SM Gibson. All right, guys, have an awesome Tuesday. I'll catch up with you tomorrow. Everyone take care.